This video is aimed at IGCSE and we're looking at the topic of enzymes. Enzymes, well, I've called these the biological supermolecules because that really is exactly what they are. All the chemical reactions that go on in your body and in life in general and any other living cell are going to be controlled by these enzymes. Let's see how they work. Well, they control reactions in cells. They control them by being biological catalysts. What's the definition of a catalyst? Well, it's a chemical which speeds up a reaction without itself being used up in that reaction. And you'll know that definition from your chemistry already. Now, they take part in the reaction, but afterwards they remain unchanged. So they are then free to go on to catalyze more reactions. So one enzyme molecule will catalyze many millions of reactions. Each cell of yours contains hundreds of different enzymes, each one of them catalyzing a different reaction. So, when you think a little bit briefly about how the activities of a cell are controlled, well, you know about the nucleus of a cell that contains the genes. It contains the genes which are made out of DNA. Now those genes in turn control the production of enzymes, and those enzymes which are controlled by the genes then catalyze the reactions in the cytoplasm. So the nucleus this way controls all the reactions going on in the cytoplasm. So when we talk about the nucleus being the control and information center of the cell, this is what we mean. It does it by controlling the enzyme production of the cell. And why are they needed? Well, all the reactions that go on in your cells would in theory be possible without enzymes, but they would just go on far, far too slowly, and so life would just be at a snail's pace. Of course, this snail itself is moving much, much faster than it would do if it were not for the enzymes that it had. Good for you, Mr. Snail. So, they're biological catalysts which speed up chemical reactions. And here's an example of such a chemical reaction. Maltose. We've heard of maltose already in our biological molecules. Maltose is a dimer. What does a dimer mean? Well, it is made out of two monomers. And in the case of maltose, those two monomers are glucose. They are two glucose molecules. So, we can convert maltose to glucose if you like. Let's change this to 2 times glucose, and that reaction is catalyzed by maltase. And you'll notice the common way that enzymes are named. We've got sugars, which very often end in O's, and enzymes which end in ase. Many enzymes end in ase, and maltase is an enzyme, you can guess by its name, that acts on maltose. That often works. It doesn't always work with enzymes, but it very often does. Oh, a little wobble there. Other key terms. The next one is substrate. The substrate is the substance on which an enzyme acts. In this case, maltose. Next word, product. This is what is made by the enzyme. And here are some examples. You'll find this table in your notes. Amylase. Amylase is a digestive enzyme. As again, we can see that A's there, and it digests starch as a substrate. Now, why is it called amylase? Well, because there's another word for starch. Let's just call it that for the moment, which is amylose. Amylose is a type of starch, and so amylase digests amylose. Indeed, it digests all starches. And what are the products? Maltose, just maltose. So we go from this, which is our polymer of polyglucose, forming our dimer, maltose. Next one, pepsin. Pepsin doesn't fit with all the normal naming strategies of enzymes, and that works on protein. Its substrate is protein, and the products are polypeptides and amino acids. Polypeptides are shorter chains of amino acids. Amino acids are the building blocks of proteins. So you take a long chain, which is a protein, and break it up into shorter chains and the monomers themselves. Lipase. You can guess what lipase works on. It works on lipids, and that produces glycerol plus 3 fatty acids. Again, you would probably guess that from your knowledge of biological molecules. Properties of enzymes and facts about enzymes, and you'll find the place to fill this in your notes. They are always proteins. Enzymes are made from protein. You need protein in your diet in order to make enzymes. Enzymes are a very important category of protein. Really, there are two categories of protein, one of which is enzymes, and the other is structural proteins. For example, your hair is made out of keratin, which is a structural protein. They're specific in their action. That means they have one job to do, and they won't do another job. And we'll look at the reasons for why in just a moment. They can be used over again. This is part of our general definition of a catalyst. They work faster at higher temperatures, although we'll put a caveat on that in a moment. They are denatured by high temperatures. By high in this case we mean typically 55 degrees centigrade and above. They're very sensitive to pH, and they work better over a larger surface area. We'll look at a number of these as we go through. How do they work? Our key phrase from this is lock and key theory. The enzyme works as a lock and the substrate works as a key, and involves something called an active site. Let's have a look at this diagram here, and there may be an appropriate place to copy this into your notes. This is our enzyme molecule here. You'll see that it has a specially shaped ridge in it, and that ridge is called the active site. The shape of this active site is extremely important. You can see that it matches the shape of the molecule of the substance. Well, we'll call that, instead of substance, we'll call that substrate. The shape, indeed, is complementary. That's an extremely important word, a very good word for using in past paper questions and indeed in real exam papers. The shape of the active site and the shape of the substrate molecule are complementary, and that's why they fit, like a lock and a key. Well, what happens when they do meet? Well, the substrate, if it happens to be moving at the right direction, remember all of these molecules are moving, they're in solution, and they're not at absolute zero, and therefore, and they're not frozen in a solid matrix, and so therefore they are moving. Now let's say this substrate molecule is moving, and it's going in this direction at just the right angle of approach as well. Well, it collides in here with the active site, it binds temporarily for a short time with the active site, and this catalyzes the reaction. This causes the reaction to occur. In this case, the substrate is being broken down by the enzyme, and so the molecule breaks at this point here, and our products are released. They are kind of bounced back out of the active site. A lot of the energy that was in this molecule when it came in and collided with the active site is then used to re-eject the product molecules from it, as well as catalyzing the reaction itself. It should be said that this is a digestion reaction here. It's a breaking down reaction. Of course, every time your body builds up a molecule, let's say it starts on amino acids and makes a protein from it, then enzymes will be doing that as well. So don't be lulled into thinking that enzymes just break things down. They don't. Some enzymes will also build things up. Now this molecule after that point will be free to take part in the next reaction. So I'm just going to put free to catalyze again. It also shows why our enzymes are specific. Let's just flick back. This substrate has this shape here, and this enzyme has this shaped active site here. If we had another substrate coming in, for example, one that just looks like this, if that tried to come into our active site here, it wouldn't fit. The shapes would not be complementary, it won't get in, and so therefore it cannot form up with the enzyme. Let's have a look at this here. So our substrates come along, these are our substrate molecules, they collide in here, in they come, maybe this one's going in this direction, that one's going in that direction, but these two happen to collide with the active site at just the right time, with just the right amount of energy, and then the enzyme helps catalyze the formation of this bond here, and our product is formed, and off it goes. And this will all happen extremely quickly. Denaturing enzymes. At high temperatures, the shape of our enzyme, or in particular the shape of the active site of the enzyme, is irreversibly changed. It's rather like one of your little ring binder files. If you throw your bag around with your ring binder file inside it, sooner or later those rings just lose their shape a little bit and then don't meet in the middle when you try to snap them together, and therefore they just don't work. Well, it's the same with enzymes. If these enzymes are being bashed around too much by being at too high a temperature and things moving around far too quickly within their particular solution, then they smash up their active site and they become denatured. Their shape changes, they become denatured, and they won't work anymore. And that also happens at extreme pH. So let's have a look at this enzyme molecule here. By the way, in reality, there is no enzyme in the world that actually looks like this geometrically designed shape here. And we have a high temperature or an extreme pH, and it just changes the shape of the protein. Proteins change shape quite easily at high temperatures and extreme pHs, and enzymes are no exception to that. We change our shape, and you can see that therefore our substrate molecule will simply not be able to fit into that active site, and it won't work. Bada boom, bada bing. Well, let's look more broadly at the effects of temperature. As you'll know from your other sciences, molecules are always on the move. They are bouncing and bumping and whizzing and spinning and doing all sorts of wonderful things like that, unless they're at absolute zero, in which case they can't. When they're in a solid, of course, they are pretty much held in position and still vibrate, and that's no good for enzymes in particular, so they need to be in a liquid situation, and inevitably that means dissolved in water. There is a depression on the enzyme called the active site that we've already talked about, and we've discussed all this lock and key theory already. Well, our next little bit to add on to this is collision theory. Now this implies that if molecules are moving more quickly, it's rather self-evident that there's a greater chance of our substrate and our active site colliding at the correct angle. And if there's a greater chance of this happening, a greater chance of a correct collision occurring, then reactions will happen faster. Kinetic theory states that at a higher temperature, molecules move more quickly, and therefore, if you raise the temperature, you are likely to get more correct collisions occurring. So, unsurprisingly, enzyme catalyzed reactions occur faster at higher temperatures, unless, of course, it's so high that the enzyme has been denatured. Typically, this works out for every 10 degrees C rise, the reaction is catalyzed twice as quickly. That also leads us onto the concept of an optimum temperature. The optimum temperature is that temperature for an enzyme at which it's working its fastest and has not yet been denatured by a high temperature. This is a graph which you will need in your notes, and let's see what it is saying. We've got reaction rate on the y-axis here, and we've got temperature on the x-axis down here. And in this case, we have no data for 9 degrees C and below. So this part of the graph, as we raise the temperature by 10 degrees C, we are approximately doubling our rate of reaction. And again, by the next 10 degrees C, we are doubling approximately our rate of reaction up to 40 degrees C. Now, much above 40 degrees C, once we get up to around this area here, we have the enzymes being denatured. Note that the enzymes do not denature at 40 degrees C. This is the optimum temperature. It is at temperatures above 40 degrees C that they begin to become denatured, and you could say really that they were completely denatured by 50 degrees C on this graph. So denaturation is complete by 50 degrees C on this graph. It does not happen at 40 degrees C. Optimum temperature varies, of course. Human amylase, such as that secreted by our pancreas or our salivary glands, denatures above around 45 degrees C. Likewise, human renin, a little enzyme we have in our stomachs to cause milk to coagulate. Very important when we're young and feeding just on milk. Denatures at 45 degrees C. Fungal renin, that denatures, well, not till 85 degrees C. Of course, if you are a bacterium in a hot water vent in the mid-Atlantic ridge, if your enzymes denature at 45 degrees C, you're not going to have much hope of survival if your ambient temperature around you is 95 degrees C. So they have very thermostable enzymes. pH. pH is another big factor which affects the shape of proteins. It changes the way that proteins bind to themselves internally, and that of course changes the shape of the protein. If you change the shape of an enzyme, then you change the shape of the active site. Now most enzymes have an optimum pH. What do we mean by optimum pH? The pH at which they catalyze reactions most quickly. Most enzymes have an optimum pH of around 7. Pepsin has an optimum pH of around 2. So pepsin is therefore adapted to work best in acidic conditions, whereas most of our enzymes are adapted best to work in neutral conditions, or indeed ever so slightly alkaline conditions. And a very extreme pH, extreme meaning a long way from its optimum pH, can permanently denature a protein. So if we take most human enzymes here at pH 7, they have their maximum reaction rate. And you take them up to pH 10 or down to pH 4, and really they're not up to very much at all. Their reaction rate is very slow. Pepsin is adapted to work at pH 2, and there it is. It has its maximum rate of catalysis at pH 2, and if you put pepsin at pH 7, it ain't going to do nothing. And that would be the end of it. Why do we have pepsin adapted to work at pH 2? Why indeed do we have a stomach at acidic pH? Well, pepsin is adapted to work in that low pH environment of the stomach. That low pH environment of the stomach is there to, well, mainly to kill bacteria. We take an awful lot of really very unknown stuff into our guts, and the stomach acid is a very effective defense mechanism against all the bacteria that might be in there, and that's a very useful defense mechanism. Surface area. Enzymes work much better if the substrate on which they're working is spread out. You spread it out and you smear it with loads of enzymes, and you're going to be doing very, very well. This is one of the reasons why we chew. We chew our food to mix it up with all the enzymes that we will secrete into it. For example, salivary amylase, and then our stomach will do a lot of churning to mix up our food in our stomach with pepsin. And this gives the pepsin a very, very large surface area over which to work. Now we're going to look specifically at the example of lipase. And lipase, as we saw earlier on, breaks lipids down into their constituent parts of glycerol and 3 fatty acids. Lipids, if you give them the chance, will form large droplets. You'll know this if you've done the washing up after a roast dinner and there's lots of fat around. You'll form quite large droplets. If the droplets are large, they will have a small total surface area. Their surface area to volume ratio will be small if they are large droplets. And this is where bile salts come in. Bile actually has 2 functions in the gut. Number 1, it's alkaline, and that neutralizes the stomach acid as the products of the stomach come out of the stomach and into the small intestine. Bile is squirted on it, neutralizing that stomach acid and allowing all the enzymes later on down the digestive system to do their work, because they will need a slightly alkaline pH to do their job. But the function we're going to think about bile here is its constituent parts of bile salts. Now these bile salts do this job: emulsification. Emulsification, very important word. And that breaks the large droplets up into small droplets, and that gives them a much, much larger surface area. Here is our beautifully drawn large oil droplet. Can you fathom how good that is? We emulsify it by bile, which is made in the liver and stored in the gallbladder and then squirted from the gallbladder into the small intestine, into the duodenum of the small intestine. And we get loads and loads of much smaller droplets produced. Now, if I were to draw around the edge of this circle, let's see how long it takes me to do it. You can sing along as well if you want. There we go. Now, how long did that take? Well, not particularly long. Now, if I am to draw around each one of these, each one of these, I'll stop singing. I'll just kind of, well, you hum along. Who knows? It takes a while, doesn't it? You can see they have a much larger total surface area than this large oil droplet. So we get many small droplets with a larger total surface area. So lipase is going to work much faster. Hooray! So this just emphasizes that point about bile containing an alkali which neutralizes the stomach acid. Why is that important? Can you answer that question? And here ends this video.